Well, there wasn't enough time during this last week before the exam for me to give a review. I thought there was going to be, but all the slots were filled with the lectures. And I know there's been kind of a demand for some type of review where we start in where we left off in the midbrain, level 10, and go up through the thalamic sections and try to cover as much as I can, especially point out the structures I think that as first-year medical students you need to know. So you should all be familiar with this series. And if you look at the mouse, I hope many of you should remember that the last section that we had in the brainstem was oriented perpendicular to the midbrain here, and went through the superior colliculus, down through the midbrain, down through the cerebral peduncle. We cannot proceed rostrally here without a good firm understanding of what's going on right here, because lots of the pathways that we left off here in the midbrain are ascending to the thalamus, and their pathways coming down through the midbrain that we're going to be able to see up in these more rostral levels. So let's take a cross-section through the ruber-duber and through the superior colliculus here, and then the next level we'll start cutting this way and go like this. So this should be familiar to you. And let me go over what I think you should know here. I think you should know that this is a superior colliculus. And this is a visual reflex center. And it relates a little bit to what Dan has been talking about in motor systems, the tectospinal tract. And that tectospinal tract would start here in the superior colliculus and cross and go down and go into the spinal cord as part of the ventromedial system. So you should at least know that the tectospinal tract is in the ventromedial system. You can see the cerebral aqueduct right here in the periaqueductal gray. And in particular for this module, there's been a lot of talk in the visual lectures about the third nerve right here. So this would be the third nucleus. The top part would be Edinger-Westphal, the bottom part would be the somatic part. And then these cells have axons which just come down like this and go out. So these would be the rootlets of cranial nerve 3. This is the big oculomotor complex. Of course, you all know the ruber-duber. You all know the nigra. And you've heard a lot about the nigra in the motor system lectures. This, I don't know how many of you remember, I've talked to some of you who called it the cerebellar peduncle, but it's really the cerebral peduncle. And what we would have coming through here would be the corticobulbars, corticospinals, corticopontines. So if we had a lesion here of these fibers, we'd have a contralateral hemiplegia, tongue would go contra, face would be weak, below the eyes contra. Uvula would be standing tall, jaw would be straight. No atrophy, of course, because this is all upper motor neuron. Then we, of course, many of you probably remember the FAB4, which would be right in here. And FAB4 would be descending hypothalamics, medial lemniscus ALS, TTT. And medial lemniscus ALS, TTT are all headed for the thalamus. And now you know where the thalamus is. It's rostral. So these pathways have to keep going. This little guy out here has his cell bodies in the inferior colliculus and headed for the medial geniculate body. You can see it over here. This is the brachium of the inferior colliculus. STT would be in here somewhere. Now, from the little bit of reviewing I've been doing with people, it's these axons right in here surrounding the ruber-duber that kind of have been forgotten. Now, you remember the superior cerebellar peduncle leaves the cerebellum, crosses into superior cerebellar peduncle. Now, these have already crossed. Now, a lot of these just keep going. So these right here are kind of going around this ruber. They're going to go all the way forward in the thalamus. In other words, they're going to be with us for a long way. They're going to go to VAVL, and they're coming from the dentate, and they're planning. So they're going to just bypass the ruber-duber, keep going to VAVL. Now, the interpositus is also sending axons out to superior cerebellar peduncle, and some of them are in here that are just going to go all the way up to VAVL. And, but a lot of them get off in the ruber-duber for a quick correction. Okay, so if If I was to ask you what these are, you could say, oh, those are cerebellothalamic fibers, John. But then I might say, gee, which deep nuclei? Then you're going to say, ooh, dentate and interpositus. Why? Because, well, that is if I told you that all these are ascending to VAVL. So keep in mind all these axons, all is Fab4, and the STT, which doesn't have its own definitive myelinated bundle, but is going to go searching for its home, which is in VPM, and that's the thalamus. Now, this is also a good slide to show you where the ventral tegmental area is, right here. Okay? Now, y'all know this is a nigra. You know these are dopaminergic, a lot of them. And there's some cells, just the more dorsal part here, that are also dopaminergic. And this is the ventral tegmental area. And, where does this project? This projects to nucleus accumbens. So that's part of our reward and reinforcement pathway. Well, how can we turn on a VTA? And I thought I'd review a bit of it. I'll review as much as I can on every slide. We go up in here, those VTA cells, so we're talking about a cell body down here. They have opiate receptors on them. So, you start drinking or you have some heroin, you turn on these opiate receptors. And there's also opiate receptors on GABA input to here. Now, think about the GABA input kind of inhibiting the VTA, but once you come in here and turn on those, these opiates latch onto the opiate receptors on these GABA neurons, are going to turn it off. So if you turn off the GABA, the VTA is going to fire a little more. These VTA neurons also have nicotinic receptors for acetylcholine. Marijuana, there's some receptors on these VTA cells too. And I think one of the most important things probably that many of you don't know, but you take these drugs too long, these VTA cells are going to shrink up and they're going to have fewer neurofilaments in it. Now, I didn't talk much about this area when I was in the brainstem with you. We did talk about one serotonergic cell group down in the medulla that was involved. Remember the periaqueductal gray projected to nucleus raphe magnus for descending modulation of pain? Well, when you think about the midline, it's kind of this Greek word for zipper. So the raphe nuclei here are serotonergic. And Dr. Banka in her sleep lectures talked some, and also in the emotion lectures where we talk about suicide and serotonin and norepi being low in depressed people, I think it's important for you to know that on the midline here, there's some cells here that are producing serotonin. And here, we go up here and we think a little bit about these raphe, this dorsal raphe nucleus in the midbrain. It's important in wakefulness, from Dr. Banka's lecture. I forgot a G here, but along with the locus coeruleus, this dorsal raphe, these serotonergic neurons and these locus coeruleus norepi neurons, they inhibit reticularis ponus, and that's the REM generator. And then somehow GABA turns off the serotonin in the raphe and the locus coeruleus, and then we kick in the REM sleep. So anyway, let's watch these fibers go forward here, okay? Here we go. Oh my gosh, I love this one right here. Well, what have we got from before? We certainly got the nigra. We certainly got the cerebral peduncle. I don't need to say anything more about that. Little piece of pons. Nigra, you know everything about that. Ruber-duber. All those pathways that we talked about are right in here. Now, the brachium of the inferior colliculus is getting off in the medial geniculate. All right? But those other ones, STT, medial lemniscus, ALS, descending autonomics, and cerebellothalamics are in here, and all these fibers here, they're headed rostrally to go to the thalamic nuclei. This is pulvinar. This is a big association nucleus. I don't think this is important for first-year medical students. This is the hippocampus, and the fornix coming off. It's actually continuous with the fornix here. Here's a fornix on the other hippocampus. How do you tell hippocampus from amygdala? Well, this is kind of coiled up, layered. When we get to the amygdala, it's just going to be a solid mass. So here we go, fornix coming up over the top. It's going to go on into the mammillary bodies. Here's the medial geniculate. This is going to go to areas 41 and 42 out here in the temporal lobe. The blood supply would be middle cerebral. Here's the lateral geniculate right here. It's got 6 layers receiving the optic tract. And these cells send their axons into this retrolenticular limb of the internal capsule. Now, the reason it's a retrolenticular limb is because it's behind the lenticular nucleus. We don't see any lenticular nucleus here. We'll see it bust in here in a couple slides. So if you had a lesion here, it's going to be a contralateral homonymous hemianopsia. No relative afferent pupil defect. If you have a whopping big lesion here, it's going to be the same thing. Contralateral homonymous hemianopia, no RAPD. So this cell goes right into the retrolenticular limb. This cell in the medial geniculate has to go rostral a little bit. These are going to go rostral to find their thalamic nuclei. Here's a nice little pineal. Here's a posterior commissure. And what do you think this is on each side? Pretectum, pretectum. Now, remember, this pretectum here can go down to this Edinger-Westphal, or it can go over to the other Edinger-Westphal. So this looks a little different than the slide we just showed, okay? But this is where your Edinger-Westphal ocular motor nucleus would be, and then it'd come out here, down here. This would be the corpus callosum, which is a big commissure. So a commissure connects 2 sides of the brain. Association bundles connect the same side of the brain. This would be the cingulate gyrus here. The cingulate gyrus gets its input from the anterior nucleus of the thalamus. Blood supply here would be anterior cerebral, middle. Down in here, classic, posterior cerebral. Thalamus, posterior cerebral. This deep bundle here, this deep bundle in the cingulate gyrus is called the cingulum. The cingulum. Well, what's different here? Well, hippo's still here, so nothing's really changed here. Peduncle's here. Retrolenticular limb is here. This is kind of, I wouldn't ask this. This is a rostral part of lateral geniculate. But this is still retrolenticular because we don't see a lenticular nucleus. Carl loves this. VPM, VPL. How do you know it's VPM? Well, it's lightly myelinated. It's hard to see, though, but that's VPM. Now, we're not going to say probably, what is this? We're going to ask you what goes here. So you're going to have to know STT and TTT. And we're going to ask you where this projects. You're going to say somatosensory cortex. How would it get there? Well, it goes through some of the posterior limbs, some of the retrolenticular limb of the internal capsule, goes up here to the 3, 1, and 2, postcentral gyrus. I asked some people, where is 3, 1, and 2? In frontal lobe, parietal lobe, temporal lobe? They didn't know. It's in the parietal lobe. Where's motor cortex? Frontal lobe. Now, don't forget, some at taste, STT is going to jump out right here. And we haven't emphasized this, but this is insular cortex. Now, something else that projects here is MD, where it brings olfaction up here. Remember, we have flavor taking place here. So this is your insular cortex right here. Okay, so what would we ask you? Hippo's good. They're better retrolenticulars, especially backed by the LGN. Is a pretty good VPM, pretty good VPL. Not a great MD. I wouldn't ask that. Corpus callosum, I don't know. I doubt it. Fornix, good fornix. Good fornix. No doubt that this is the fornix because it's attached to the hippocampus. Now, you know, when we get in front of the LGB, we're going to see the optic tract, and you're going to say, oh, is that the optic tract or is it the fornix? Well, this is really attached here to the hippocampus. So this is continuous, okay? So this is a good place to ask it. So let's see what the next one. So VPM, VPL, those are the hardest ones. You see a little piece of putamen busted in here, but it probably busted in right here. I bet if I leave my pointer right there, it'll be a putamen here. Let me see. Ooh, almost. Putamen right here. Caudate. And something going underneath the putamen. Sublenticular. So if I said, gee, where did he start? You'd say, medial geniculate body. If I said, where does it end? You'd say up here in 41, 42. If I asked you the blood supply for 41, 42, you'd have to say middle cerebral. Okay, sublenticular. Now, this is kind of hard. Still hippocampus, but this is optic tract. How do I know that? Well, we're rostral to the lateral geniculate, and, you know, there's a little separation here. I probably wouldn't ask it on this one. Okay, but I'd ask hippocampus. I'd certainly ask the nigra, and you know the nigra, no matter how you cut it, always sits on the what? On the peduncle. So here's the posterior limb of the internal capsule, feeding into the peduncle. Here's the sublenticular, really good. Optic tract. I could say, gee, what's the visual field loss with an optic tract lesion? You say, oh, contralateral, homonymous, hemianopsia, or anopia. Would you have a relative afferent pupil defect? No. Gee, where does the optic tract go? Lateral geniculate body. Gee, where does it start? The retina. Well, on the ipsi, is it the temporal retina or the nasal retina? On the ipsi, darn this thing. On the ipsi, be the temporal retina. On the opposite eye, be the nasal retina. Then I could push you further, and I say, what gives rise to these axons? Photoreceptors, bipolars, amacrines, horizontals, or ganglion cells? And the answer is ganglion cells. A lot of kids didn't know that this week. Anyway, the next thing I could say is, oh my gosh, what's the blood supply to the upper part of the posterior limb versus the lower part of the posterior limb? Are you going to say lateral striates up here, anterior choroidal down here? Double jeopardy. If I said, gee, what's the blood supply to sublenticular and retro? You're going to say anterior choroidal again. So lateral striates, anterior choroidal. Now, the nigra always sits on a peduncle no matter what. Here's the ruber-duber. Here's some VPM that's gotten actually a little bigger. Okay? Here's a VPL. So you got all your pathways in there. Some of them are getting off in here. Some of them are getting off in here. And some of them are going to go to VAVL. Don't miss this one right here. Now, first of all, it's not VPM or VPL. They're up here. It's not the nigra because the nigra sits on the peduncle. It's the subthalamic nucleus. Subthalamic nucleus. It's, is he going to ask me that? Oh, probably not. Probably have a little pointer here and I'll say, gee, what happens if I stimulate that? Would I get more or less movement? And you're going to say, gee, I'm going to get less movement because it's going to excite GPI. I say, oh my gosh, classical national board question is going to be, what if I have a lesion in here? Oh, you lose that excitation of GPI and you have hemibolism. And I'm going to say you're going to have the hemibolism on the same side as the lesion on the opposite side, and you're going to say the opposite side. Then I'm going to say, what are you going to give this patient? A cholinergic agonist or a cholinergic antagonist? You're going to say, gee, what pathway is dominating, Dr. Harding? The direct pathway. Well, then I'm going to give it a cholinergic agonist. So that's how you have to think about it. VPM, VPL, subthalamic. I like subthalamic here better than the next slide. MD, good one. Always going to be here on the midline. A big dude, you know, pretty big. Only midline nuclear group you got to think about. It's unique and there's a thalamic nucleus input from the olfactory cortex. Going to then send that olfactory information to orbital frontal cortex. Going to send some of the olfactory information out here in the insular cortex. Also get some pain fibers. Goes to prefrontal cortex. Now, as far as this stuff, forget it. I'm not going to ask you this. The next thing after VPM and VPL I'm going to ask you out here is going to be VAVL. MD, of course, you got to know. Subthalamic, you got to know. You know the fornix, you know the optic tract. This optic tract is going to get closer and closer together as we go forward. Ooh, now, here'd be the optic tract. This one I don't like either because some poor student will say, here's the hippocampus because Harding said it's coiled and layered. And there it is. Oh, it could be the fornix. Well, it's the optic tract. Now, over here, this is going under. Here's globus pallidus putamen. This is going under again, so this is still sublenticular. That's kind of tricky there. What always sits on the peduncle is the nigra. So that means this is a nigra and this is subthalamic. Let's show it better over here. This would be the cerebral peduncle. Here's your posterior limb leading into the cerebral peduncle. This next clear area is a nucleus. That's nigra. And then kind of this area would be subthalamic. I like the other one better. MD, not going to worry about this. Optic tract is a little better here. Might ask you there. Hippocampus, not amygdala. Not amygdala. But this is the hypothalamus. This is the hypothalamus. So what do we know about it? Well, we know that this is the target of the fornix. We know that there are cells in here that give rise to mammothalamic. We can't see it, but it's going to go up this way to the VAVL. This isn't VAVL, this is MD, but fornix is going to be coming in here, MTT is going to be leaving this. Now we're in the kind of in the posterior hypothalamus. It's a good time to talk a little bit about sleepy time, a little bit about sleep, because in that drawing that we put together, this posterior hypothalamus, it's got a couple things going on that are related to sleep. And, well, why don't we take what we know about this? A big lesion here is going to give you hypo, H-Y-P-O, thermia. But also there's cells in this posterior hypothalamus involved in sleep. One of them is a tuberomammillary nucleus. You can't identify it, but it produces histamine, and that's really alerting, turns you on. Other cells in this posterior hypothalamic area send fibers to cholinergic neurons in nucleus basalis minor. Okay, so it can turn on nucleus basalis minor. Now, don't forget, nucleus basalis minor is further far forward, and it's got cholinergic neurons then that kind of play a role in alerting or, wakefulness in, by basalis sending a projecting cortex. But also don't forget that there are some neurons in nucleus basalis that are GABAergic and play a role in non-REM sleep. There are also other cells back here in the posterior hypothalamus that are cholinergic, and they project to the thalamus and then up into cortex, so it's alerting. This is just, I'm trying to integrate this area with what Carl taught you and what Ruth Benka taught you. Okay? Let's go on to the next slide here. Oh, amygdala. Amygdala. How do I know? Well, this is the almond. I mean, it looks like an almond to me, but it doesn't look like the hippocampus to me. You go over here, this is weird looking, right? Because this is kind of little something that looks like the fornix. So I'm going to ask you over here. You can't confuse it with the hippocampus. It's rostral to it, and it's a very solid mass here, okay? Where's that thing? Right there. Now, don't miss this. Don't call this the hippocampus. What else could you call it? Nothing. Right here, optic tract. Very good. This is cricket. This is not the hippocampus, so this couldn't be the fornix. This is the optic tract. Now, I like this area. This is the tuberal region. This is the tuberal region of the hypothalamus. Now, here's a bundle, here's a bundle, here's a bundle, there's a bundle, okay? Now, I always say when you got 4, 1, 2, 3, 4, the bottom one is fornix, the top one is mammillothalamic. Now, this fornix is actually running caudally to go to the mammillary body, which we just looked at. This MTT is running kind of rostrally and dorsally to go up into the anterior nuclei, okay? Now, this fornix kind of is a good landmark. Here is the ventromedial nucleus where the rat's going to eat too much and be real nasty. Here's the lateral hypothalamic area where it's going to be anorexia. And down in here would be arcuate nucleus, and my key word there would be releasing hormones. This is some MD right here, okay? Now, here's your posterior limb. We already talked about the blood supply up here, the blood supply down here. And you remember there's two ways out of the GPI. One of them just busts right through the posterior limb. Just goes right through it, invades it. Goes right like that, and then hooks around and goes forward to VAVL. This is the lenticular fasciculus. Okay? So we haven't emphasized it much. I probably won't ask you on the exam, but for some of those you interested, cells in here go right through the posterior limb here, and then we can't see it, but eventually it'll hook around and then go forward. So what would you have in this little bundle right here? You'd have all those cerebellar thalamic fibers, right? Because we're in front of VPM and VPL. So cerebellar thalamic fibers would be here, and some of the lenticular fasciculus fibers are in here also. But you can't actually see the connection. So this would be lenticular fasciculus. If I ask you what the cell bodies are, you'd say GPI. If I ask you where it ends, you'd say VAVL. Optic tract, going to get closer together. Amygdala. Now, over here I've listed stuff about the amygdala because I think you've probably forgotten it. But, when I think about the amygdala, I think about fear, and I think about it really turning on, and I'm getting anxious, and my sympathetic system's going up, and my cortisol's going up. And all these connections here, all these connections enable me to get real hyper and enable my cortisol to go up and enable me to, my hair to stand up. And why? This projects, first of all, there's cells in here. There are actual cells in this nucleus that have CRH. So all this CRH is not produced by the paraventricular nucleus, but there are some extra hypothalamic, extra outside hypothalamic neurons like the amygdala have CRH in them. And the amygdala is going to send the CRH down to locus coeruleus and turn it on. When you turn on the locus coeruleus, you get all that norepi floating all over the place. Okay? Now, when you have bilateral lesions in here, calcifications in here, we have Urbach-Wiethe disease. And I hope you know that you can't tell the intensity of fear in people's faces. It projects to PVN, we just said that. It projects to the hypothalamus. Remember when we talked about in brainstem that descending hypothalamic pathway that goes with the ALS in the brainstem and goes down with the LCST in the spinal cord that turns on T1 to L2? Well, this will go to the hypothalamus, and then you go down and turn on that sympathetic system. The amygdala also goes to VTA, so you put out more dopamine, which is very alerting. Goes to locus coeruleus, which we talked about. It's going to go to the trigeminal and facial nuclei because think about a fearful face, you know, and a clenched jaw, okay? And all the outputs, all the cells that are projecting out of this thing are in the central nucleus of the amygdala. Now, there are lots of inputs to this, but just think if you have a memory, memories are going to go flying from the hippo and cortex into amygdala. Cortex, prefrontal cortex is going to go there. All the inputs, the afferents to the amygdala, go to the lateral nucleus, and then from the lateral nucleus can go right to the central nucleus or to these other two nuclei, which in turn go to the central. So those are kind of nitpicky things, but I thought I'd give you a chance just to review a little bit with me. All the amygdala right here. Now, this will still be the amygdala, but we got the jug handle. That's key here. Probably ask you this. It's really great, I think. It's going from GPI to VAVL. Now, when you see the jug handle, no doubt about it, I can ask you VAVL. Because anything that goes into this, you know the jug handle or the ancilla lenticularis goes to VAVL. So I could ask you this. This is different than the lenticular fasciculus because look, it scoots around underneath the posterior limb. The lenticular fasciculus barged right through it. I've been asking some students I've been reviewing with, what happens if I cut the ansa? Would you have more or less motor activity? Well, if you cut this inhibitory input, you're going to have more. If you stimulate it, you're going to have less. VAVL, I asked some people, how do these cells get to cortex? Well, one of them said, gee, you know, it kind of swims up to the lateral ventricle, barges through the cortex. No, it goes in the posterior limb of the internal capsule. It's right here. It's very convenient. This is a two-way street. Upper part, lateral striate, ventral part, anterior choroidal. Here's your putamen, globus pallidus, amygdala. This is a little piece of the anterior commissure. It's mentioned in Carl's lecture on olfaction. It'll get closer. We'll watch it. It's going to get closer and eventually cross. Optic tract, this is the best place. I love it. It's not the fornix. You know it's not the fornix because this is a fornix. Remember? 1, 2, 3, 4. The bottom two are fornix. The upper two are mammillothalamic. If this is mammillothalamic, then I can ask this. This is the anterior nucleus because you know the mammillothalamic goes to the anterior nucleus. What better way to know it? It's going right in there. This is the anterior nucleus. So how does it get up to cingulate cortex? Jumps in the posterior limb, goes right here. Very convenient, very convenient. Blood supply to the thalamus, branches the posterior cerebrum. Blood supply to the upper part here, lateral striates, bottom part anterior choroidal. Would you be skinny or would you gain weight with a lesion here? Oh, you're going to gain weight and you're going to be very, very, very nasty. Out here, you're going to be thin, can't really see it, arcuate down here. All right? But here's the jug handle, right there. So this is VAVL. Caudate, corpus callosum. All right. Ooh, optic chiasm. Rostral or anterior. Rostral and anterior are the same thing. Hypothalamus. Hypothalamus. Now you can see the jug handle here. All right? So you can see it going up, so it's got to be VAVL. Anterior, you might not know. You say, oh, that's MD. No. This is anterior. It causes a little bulge here in the lateral ventricle here. This is anterior. That might be kind of tricky. Posterior limb, we know the blood supply. Why is it posterior limb? You got thalamus. Anytime you got the thalamus, you got the posterior limb. Here's the anterior commissure getting closer together. Here's the optic chiasm. Lesion right down the middle, bitemporal hemianopia. Relative afferent pupil defect? No, no, no way. Fornix, because you don't see any mammillothalamic tract. Remember, if you got 4, the bottom one's fornix. If you got 2, like 1, 2, it's fornix. Paraventricular nucleus, superoptic, suprachiasmatic, circadian rhythms, oxytocin, vasopressin, CRH, oxytocin, vasopressin. This might be a little piece of the uncus here, okay, uncus. Caudate, fornix. I bet you don't know what this is. Yeah, I don't know. This is septum pellucidum. Septum pellucidum. Here's your corpus callosum. Here's your cingulate gyrus. Now, this rostral or anterior hypothalamus also is a part of the sleepy time. Well, first of all, you got nucleus basalis here. I don't know how many of you can identify nucleus basalis. This is the one that really dies in Alzheimer's. So you lose all this acetylcholine that's going to cortex. But don't forget, this has acetylcholine, cholinergic neurons. It's got some GABAergic neurons. Don't forget this. So this contributes to non-REM sleep. And you know, Alzheimer's patients have lots of problems with their sleep. Now this rostral hypothalamus, no particular region in here. No particular. We're just going to say from this area. There are some cells up here in this hypothalamus that contain GABA, and they project to cortex and thalamus, so mainly the cortex, so they're going to play a part in non-REM sleep. So anterior hypothalamus, GABA, non-REM sleep. Now, a lot of you are probably asking where the preoptic area is. The preoptic area would be one little tiny slice in front of this, rostral to this. And what's in the preoptic area that all good M1 should know would be VLPO, VILPO, okay? You know, the adenosine goes up in these cells, and all of a sudden the GABA kicks in and goes down and inhibits lots of brainstem nuclei, okay? Don't forget that for non-REM sleep. Okay, we're almost done. Ooh, is this the olfactory bulb tract or optic nerve? This is the optic nerve right here. If you had a lesion here, you would certainly have a relative afferent pupil, okay? Now, I should have talked a little bit about that with cranial nerve 3, but I didn't. But this is, boy, you get this, you're going to have an RAPD. You get more than half the retina, you're going to have an RAPD. So just don't miss those questions. I asked some people what they thought this was, and nobody got that right, but it's part of the olfactory tract, not the nerve. The nerve is probably lying in the ethmoid bone, cribriform plate. Here's your anterior commissure. It just got, you know, I don't think I'll ask you that. Fornix, I might. Septum pellucidum, corpus callosum. Now, putamen, caudate, globus pallidus. This is a septal area. We haven't talked at all about it, just so don't worry about it. This is not the gateway to the cortex. This is not the thalamus. If you don't have thalamus, you get anterior limb, anterior limb, anterior limb. Blood supply? Medial striates, which are branches of the anterior cerebral. That's kind of picky. That's kind of picky. And then this one, nucleus accumbens. That's a good one to ask. Caudate, putamen, this is something special. We call this the ventral striatum. This is where the VTA projects and dumps out all that dopamine for euphoria, and then that animal gets real active, okay? This is nucleus accumbens. And this is where the dopamine, you know, boy, these, you know, if you're taking cocaine or amphetamines, there's a heck of a lot of dopamine up here, more than normal. Why? Because cocaine and amphetamines, they tie up that dopamine transporter. And by tying up or plugging it up, you have more dopamine around. So don't forget that. That's really important. As is this one. As is this one. So when I look at this, I look up here, I don't know what this is. P. I don't know. I couldn't get that off in my PowerPoint. I kept trying to get that off. This is rostral, caudal. This is visual cortex. If I said, what's the blood supply to this? In here especially, you're going to say posterior cerebral. Yes. Then I'm going to say, what's this? Ooh. Well, it's white matter, and it's behind the lenticular. It's retrolenticular, area 17. Where are the cell bodies of these? Lateral geniculate body for the most part. Where are these ending? Area 17. Now, I don't know if I'm in the upper bank of the calcarine. I don't know if I'm in the lower bank of the calcarine, but I know that central vision's back here and peripheral vision's up here. You know that from the practice questions. Okay. Now, the next thing I say, is this a caudate? Or is this? Well, the caudate and the putamen are the striatum, and they stain the same, right? So this is caudate, this is putamen, and this is thalamus, okay? So the spatial relationship that you got to keep in mind is the caudate and the thalamus are medial to the internal capsule. Here's the posterior limb, here's the genu, cortical bulbars, here's the anterior limb. But caudate and thalamus are medial. Lenticular is always lateral. No matter how you cut the brain, you could cut this thing schlonz-wise, and caudate and thalamus are going to be medial, and lenticular is going to be lateral. I don't know how you cut it. Most important pathway in here, corticospinals. Also some ascending things from VPL and VPM, but they're pretty diffusely distributed. This is National Board number 1, cortical bulbars. Remember, the posterior limb gets a dual blood supply depending upon where we are in it, lateral striate or anterior choroidal. This is anterior choroidal. Now, somebody asked me this week, where is sublenticular? Well, it'd be underneath this, so we wouldn't see it. We'd have to cut lower. The ganu got those cortical bulbars in it, and supplied by the lateral striates, okay? And then this anterior limb, I don't know, it's got some corticothalamics, some thalamocorticals, and it's got some corticopontines. But, you know, like Carl said, it's not that important clinically. This one is, and this one is, and of course this is. So that'll do it. I could go back through and ask you the hardest question, but and people get mad if I don't give the answer. So, oops, I will do that though. But I'm going to tell you the answer. What limb is this? Are you ready? Anterior. Would you have a relative afferent pupil defect with a lesion here? Yes. Would you have a relative afferent pupil defect with a lesion here? No. Is this supraoptic or suprachiasmatic? Supraoptic. Is this the lenticular fasciculus or the ansa? The ansa. Is this the optic tract of the fornix? Optic tract. Oops. Am I going to gain weight or lose weight with a lesion here? Well, I'm going to gain weight. Am I going to be nicer or am I going to be grumpier? I'm going to be grumpier. If I had a bilateral lesion in here, could I have a fear response? If a rat had a bilateral lesion in here, could it freeze? No, it can't freeze. It can't have a fear response. Ooh, hypothermia or hyperthermia? Ooh, hypo. Too much movement, too little movement? Too much. Too much. Lesion here, too much or too little? Too little. Is this the optic tract? Yep, but I probably won't ask it because it's too close to the hippo. I could ask this. What is this? Sublenticular. Very good, very good, very good. Here, would I ask the rostral pole, the lateral geniculate? No, but I'd ask the fornix. Why would I ask the fornix? Because I can see it coming off the hippo. Would I ask VPM? Yeah. Would I ask VPL? Yeah. Would I ask MGB? Yeah. Would I ask LGB? Yeah, because I love Napoleon's hat. Would I ask retrolenticular? Yeah. Would I ask you the blood supply? Probably not. Would I ask you the ruber-duber? Yes, because you know I love it. Would I ask you the pineal? No. Would I ask you pulvinar, the cushion? No. Would I ask you the pons? No. Would I ask you ruber-duber? Oh, would I ask you VTA? I mean like an initial or Weiger? Probably not. It's too hard to pick out. Would I ask you the brachium? No, that was before. Would I ask you the Fab4? No, that was you already been tested on that. Would I ask you what happens if you have bilateral lesions of the superior colliculus? Paranoid syndrome. Paranoid syndrome. Yep, I might ask you that just because I like paranoid syndrome. Can't elevate your eyes. Now, if I had a little lesion here, what would my, if I had a lesion here, say this is the left side, my pupil would be very big. But if you're in dim light, the other one's going to be pretty big too, so they're going to be symmetrical. All right? This pupil is never going to move no matter where you swing that flashlight if this is the only lesion. This is an efferent peduncle. Efferent lesion, sorry, not peduncle. Here's your cerebral peduncle, okay? Oh, let's go back to this one here. I know you hate this, but that's all right. I got a lesion here on the left, okay? Where's your hemiplegia? Ah, there isn't one. Which way does your tongue go? Contra. Any atrophy? No. Any fasciculations? No. Which way does your uvula go? It doesn't. Which way does your jaw go? It doesn't. Where's your face weak? Contra. Below the eyes. Last time I'm ever going to tell you that. So have a good time with this. You can watch